Good afternoon. I'm Dr. Brian Shapiro, a scientific content specialist at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled Making Sense Out of Microbiome Data, the Importance of Standards, presented by Ms. Briana Benton. Ms. Benton is a technical manager at ATCC. In this presentation, Ms. Benton will provide an overview on the current challenges in microbiome research and will demonstrate the application of mock microbial communities in assay development and standardization. A special focus will be placed on ATCC's wide range of fully sequenced and quantitative mock microbial communities that support the development and optimization of microbiome research workflows. If you have any questions for our speaker, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. So with that, I would like to welcome Briana Benton.
Thank you, Brian, and thank you to the audience for joining our webinar. Today, I would like to introduce you to ATCC's continuing effort in the development of microbiome standards. For some of you in the audience, this may be a refresher, but for many of you, I hope this opens up a dialogue about the undeniable need for standards in microbiome research. I will discuss the numerous challenges with microbiome research. These are well-documented challenges that should really drive home the undeniable need for researchers to include standards in all aspects of their research. I will also show our current portfolio of microbiome standards, including a few new additions, and lastly, I'd like to share some application data. My intent is not to show the best data. In fact, I would like to highlight some of the challenges that we've experienced with extraction and NGS library preparation methods, to name a few. Briefly, a little background on the American Type Culture Collection, or ATCC. ATCC was founded in 1925 and is a private, independent, nonprofit organization headquartered in Manassas, Virginia. We also have additional R&D facility in Gaithersburg, Maryland. ATCC is the world's premier biological materials resource and standards development organization. We collaborate with and support the scientific community with industry standard products and innovative solutions. Our broad range of products include cell lines, microorganisms, genomic and synthetic nucleic acids, and media and reagents, and of course, the focus of today's talk, are microbiome standards. Each of our biomaterials are expertly authenticated and characterized using genotypic, phenotypic, and functional analysis. So why all the interest in microbiome? Well, the area of microbiome research has rapidly evolved over the last decade. A recent PubMed Central search revealed over 58,000 hits, and a simple Google search produced over 11 million hits. The point is, microbiome is generating a lot of interest, and not only from the scientific community. As the topic gains more and more momentum and the general public looks to their own microbiome for answers, we as a scientific community need to be in the position to provide data that was produced with sound quality principles and practices. Unfortunately, that has not always been the case. For years, publication of microbiome data without the use of controls was accepted, and that needs to change. As a world-renowned standards organization, ATCC is committed to addressing this need. We have devoted a lot of time and resources to addressing how we as a standards organization can help our customers with their microbiome research. Our response to this growing need was to research, develop, produce, and distribute whole cell and genomic DNA mock microbial communities, which leads me to the important question. Why are standards so important? Because microbiome research is extremely challenging due to the inherent struggle of using one method to collect, preserve, extract, and prepare and analyze what may potentially be a very diverse sample set. And what do I mean by this? Well, it is one thing to optimize a workflow for a single organism, but it is quite a different scenario when one tries to apply a singular method to a diverse sample set. Here you can see an example of a metagenomics workflow, and you can see where bias can be introduced at any stage of the process, from sample collection all the way through data analysis. These biases can obscure the true composition of a microbial community and lead to inaccurate analyses and incorrect conclusions. Standardization of these different types is challenging without relevant controls. The ATCC microbiome standards are intended to provide researchers with a mock microbial community of well-characterized organisms that are present in known concentrations. It is with this knowledge that researchers are in a better position to assess the efficacy and reproducibility of their respective workflows and to identify any potential issues that may impact their data. It is only after a bias has been identified that a corrective action can be considered. It is important to realize that there is no perfect microbiome workflow. The goal here is to gain a better understanding of the challenges and then address the challenges once they are identified. So how does ATCC choose the strains for inclusion in a mock microbial community? We take into consideration a lot of variables which range from cell wall properties to genome complexity and size, 16S ribosomal RNA copy number, strain relevance, and of course the availability of published reference sequences. We also seek input for customers and key opinion leaders in the field. This is not an exhaustive list of requirements, but it does illustrate the diversity and complexity of the isolates chosen for inclusion in ATCC microbiome standards. It is important to remember that the microbiome standards are meant to mimic native microbiomes in complexity, not in the quantity of organisms. ATCC microbiomes are available in two formats. The first format is a whole cell product which consists of authenticated ATCC cultures mixed in even proportions and shipped at room temperature. The whole cell controls are an ideal full process control because they can be incorporated into the very beginning of a workflow and processed just as an unknown sample would be processed. The second format is our genomic mix, which contain nucleic acids prepared from authenticated ATCC cultures. Each individual component is then extracted using the most appropriate method for that organism. And for an added challenge, the genomic mixes are available in both even and staggered genome concentrations. The genomic mixes are ideal for assay optimization, library preparation and validation, just to name a few. Think of these as a mid-process control. So what does ATCC offer? This table illustrates our generic standards. The first four standards are mixtures of genomic DNA prepared from 10 to 20 diverse bacterial species, mixed in even or staggered proportions. The most challenging of the set would be MSA 1003, which includes genomic DNA from 20 different species in staggered proportions. The staggered proportions of organisms, some of which are in very low concentration, can help researchers assess their limit of detection capabilities. Next, we offer our whole cell products in even concentration. As mentioned earlier, the whole cell products are ideal as a full process control. For clinical metagenomics applications, we work closely with our LGC collaborator to offer MSA 4000, which is a mixture of genomic DNA from blood-borne pathogenic bacteria that is mixed in staggered proportions. Additionally, the components of MSA 4000 are quantified with ddPCR, which makes MSA 4000 a great tool for NGS-based pathogen detection. Lastly, we collaborated with our ABF Metagenomics research group led by Scott Tye from the University of Vermont. Under their Extreme Microbiome project, they created 3 genomic DNA standards that contain environmentally important diverse bacteria, including extremophiles. These controls can be used to assess library preparation methods, run-to-run variability, and many others. All these general standards are available in the ATCC catalog and support different aspects of microbiome research, such as assay development, LOD testing, pathogen detection, and environmental testing. Just this past year, we debuted our site-specific standards for research on the oral, skin, gut, and vaginal microbiomes. These are set up similarly to our generic microbiomes. They are offered in whole cell and genomic formats and comprise strains that are prevalent in the respective body site during normal and disease states. The whole cell products are offered at 2 × 10⁷ cells per organism, and the genomic products are offered at 2 × 10⁷ genome copies per organism. The whole cell products arise in a lyophilized format. Simply rehydrate the pellet using caution to ensure the entire pellet is rehydrated and homogeneous, aliquot the material, repellet, and freeze the unused aliquots for later use. Recently, we launched our Virome standards. These standards are offered in both the whole virus and nucleic acid formats. Standards are prepared from genetically diverse, clinically relevant, well-characterized viruses that were selected on the basis of genomic organization, envelope properties, and genome size. We have included enveloped and non-enveloped DNA viruses, such as the herpes and adenoviruses, mixed with 4 RNA viruses. the positive sense Zika and the negative sense influenza and RSV, and lastly, the double-stranded reovirus. Additionally, the quantity of each component is confirmed via ddPCR. The application of the Virome standards is to provide researchers with a standard to optimize their sequencing methods and to recover sequences from diverse viral families. And lastly, we have our spike and whole cell and genomic standards, identified as MSA 2014 and MSA 1014, respectively. This product line comprises 3 fully characterized and authenticated ATCC organisms that contain unique synthetic 16S tags incorporated into their bacterial genome. I'm going to discuss this product line in a bit more detail because they are different than our typical microbiome product line. These products can be used as a true internal control, or a spike-in, if you will. We created 3 unique synthetic 16S tags mimicking the native 16S ribosomal RNA gene from 3 bacterial strains, E. coli, C. perfringens, and S. aureus. Each tag consists of 4 artificial variable regions corresponding to V1 through V4 of the 16S ribosomal RNA gene, a unique synthetic sequence, and the conserved 16S regions for PCR amplification. To be clear, the native 16S genes were left intact, and an artificial 16S sequence with a synthetic tag was incorporated in each respective genome. Each organism contains only 1 copy of the synthetic tag. The genomic and whole cell 3-strain tag standards can be used in the same manner as all the other microbiomes I have previously discussed. However, this product line is intended to be used as an internal control. Additionally, these products are quantified via ddPCR. Researchers can add a known copy number to an unknown sample and use this known copy number to normalize data within a sequencing assay and even between different assays. Here's an example of 2 scenarios. Sample X is spiked with 100 cells carrying the synthetic tags. This results in a relative abundance of 0.99%. Knowing the 100 cells equals an abundance of 0.99%, one can extrapolate that 19.8% equals 2,000 cells, or 59.41% equals 6,000 cells. For more information on our 3-strain tagged whole cell even mix, simply navigate to the product page, click the Documentation tab, and click on the Technical Data Sheet. Our genomic mix, MSA 1014, is scheduled to launch 30th of August 2019. Most of the initial studies on the human microbiome have primarily focused on bacterial communities, but recent advancements in sequencing technologies have enabled the community profiling of fungi. However, the complexities associated with metagenomic sequencing analysis pose significant challenges toward assay standardization. To address this, we have developed 2 fungal mock community standards consisting of 10 diverse and clinically relevant fungi. Currently in production, we have a whole cell and genomic Mycobiome Standards, which are scheduled to launch in the fall of 2019. The Mycobiome products were developed with similar requirements as our earlier products. The whole cell products consist of authenticated ATCC cultures mixed in even proportions in a lyophilized pellet, which are ideal as a full process control. The second product format is our genomic mix again, which contains nucleic acids from authenticated ATCC cultures. Each individual component is extracted using the most appropriate method for each organism and then mixed in even genome proportions. The fungal mock community standards are ideal for assay development, optimization, verification and validation, evaluating reproducibility, and even use as a daily run control. So with that is the conclusion of ATCC's current and soon to launch Microbiome, Virome, and Mycobiome standards. And with that, I'd like to reiterate the importance of using known and trusted standards in all aspects of microbiome workflows, whether it is a whole cell standard, handled and stored and extracted alongside an unknown sample, or if it is a genomic standard used to optimize primers or library preparation methods. Now let's move on to some application data. Customers often ask us for recommendations regarding reagents, such as which are the best extraction kits or library preparation kits. These are just to name a few. The short answer is, we don't recommend specific kits or methods, but instead encourage customers to use the standards as they would an unknown sample and then evaluate the data. When evaluating the data, 2 questions must be asked. Were all the organisms detected, and were they detected at an acceptable abundance? If one or both of those answers is no, then an evaluation of one or more steps is required. Here I will show some application data to demonstrate how microbiome samples can be used, microbiome standards can be used to evaluate different aspects of a workflow, whether it be DNA extraction, 16S sequencing, NGS library kits and platforms, and lastly, bioinformatics. I will start off discussing extraction methods because it seems to get the most attention and is often the easiest to blame when results are not as expected. In the top illustration, you can see the genomic mix. In this case, MSA 1004 is fairly consistent for an even mix of organisms. The blue bars represent the theoretical expected abundance, and the orange bars represent the actual run data. And you can see that the genomic mix is fairly consistent with the expected range. However, the scenario is very different when the whole cell mix, which contains the same organisms as the genomic mix, is extracted with two different commercially available kits. This data shown on the bottom highlights the true struggle with DNA extraction of microbiome samples. With kit A, shown in orange, we recovered less actinomyces than expected. Using kit B, shown here in gray, the recovery of S- was less than expected. Not surprisingly, both gram-positive organisms. So now what? We decided to take a much closer look at what was happening with the individual organisms within MSA 2004. We took each of the organisms and extracted them individually with the same kit A and kit B as previously mentioned. Each organism was normalized to approximately 2 × 10⁷ cells and then extracted. Surprisingly, when we factored in the concentration of the recovered DNA with the recovered volume and then normalized them for genome size, the recovery of each organism produced an overall average yield of about 1 × 10⁷ genome copies per organism. This truly illustrates the complicated nature of microbiome research. When a particular extraction method works reasonably well on a singular organism, but when the same organism is mixed with other organisms, that same kit may not perform as efficiently. Unfortunately, this doesn't help formulate a conclusive decision on what is the best extraction kit, but it does provide further evidence of data bias. Here we extracted the whole cell gut microbiome standard, which is an even proportion mix of 12 organisms, and evaluated the NGS and 16S ribosomal RNA community profiling on an Illumina platform. For the shotgun analysis, all components were identified with the observed ratios were slightly skewed compared to the expected ratios. It is important to remember this is a common finding when analyzing whole cell standards. Even when starting with a validated standard, no one method will result in perfect data during final analysis. Additionally, we evaluated the 16S V1, V2 region, and this is what one would expect to see for 16S profiling on a microbiome sample. It is difficult to draw definitive conclusions from 16S data because not all primer sets have the same affinity per organism. Additionally, the shorter sequencing reads coupled with the highly conserved regions within the 16S gene lead to nonspecific identification. These two factors lead me to the next slide data. Here we evaluated the oral microbiome genomic mix with 2 different universal primer sets, which amplified different regions within the 16S ribosomal RNA gene. As you can see, although the results are not drastically different, a potential for bias does exist due to the choice of primers and PCR amplification. One way to address this inherent bias of 16S amplification is to sequence the entire 16S gene with true long-read sequencing, or a modified method, such as loop-seq. Using the loop-seq 16S sequencing method, which produces a synthetic long-read, our 20-strain mixed proportion genomic standard produced really exciting results. For reference, the column on the left illustrates the expected abundances. The middle column illustrates the results of the loop-seq method. The reported abundances were virtually spot on with the expected abundances. This is because the long reads removed the inherent biases typical of 16S sequencing and resulted in 0 false positives. However, the column on the right illustrates the results of 16S sequencing with the V1, V2 primer set, which produced data that was less than ideal when compared with the full gene sequencing produced using the loop-seq method. And as you can see, it is difficult to reach species-level identification using the conventional 16S sequencing method. Now, before I jump into the comparison of library kits, I want to revisit one of the earlier topics when I discussed the selection of microbiome components, specifically the GC content of each potential candidate. This is an example of the oral microbiome, and I want to draw your attention to the two organisms that have vastly different GC content. This will be important for our next slide. In collaboration with Illumina, MSA 1004, which was shown on the previous slide, was prepared with the Nextera XT kit shown on top. Now this data is not ideal because this is a genomic mix. In other words, the extraction bias should not be an issue. So one should expect to see a more even distribution. But when the same mix is prepared using the Nextera Flex kit, the results are far more in line with expectations. Now, notice the difference in the representation of the Fusobacterium actinomyces between the XT and Flex kits. This advancement was really exciting to see and further supports our effort to encourage the use of standards in every step of the process to include evaluating new technologies, kits, and methodologies. Now let's take a look at NGS platforms. We have demonstrated some of the variability in extraction methods, 16S primer choice, and even library preparation kits. But now, in collaboration with UIC, we evaluated the 10-strain staggered MSA 3001 and evaluated the 4 different Illumina platforms. You can see the consistency in the data between the 4 different platforms. In addition to different Illumina platforms, MSA 1003 was evaluated on the Ion Torrent platform. All components were detected for both the NGS and 16S runs, but you can see a slight variability between the expected and NGS results, and more specifically, the slight variability between this platform and the previous slide. So let's move on to some long-read platforms. Here we compare 2 of our staggered mixes on Oxford Nanopore Technologies and PacBio platforms. In the graph on the left, we compared our 10-strain staggered mix on the Illumina MiSeq and ONT MinION. We saw a slightly unusual proportion of reads for 2 of the components when we used the ONT platform. But in general, all components were identified and the proportions are within an acceptable range. The graph on the right shows a comparison of our 20-strain staggered mix on the Illumina and PacBio platforms. All components were identified and the abundances are clearly within an acceptable range for both instruments. During the past year, we introduced many of our standards onto the ONT Nanopore sequencing platform. Here we illustrate MSA 1006 after 60 minutes of runtime. For those of you familiar with ONT, this data was generated with the 1D ligation kit. We see here a slightly higher variability in the number of reads generated per organism. For example, Bifidobacterium was lower than expected, but overall, all 12 components were identified. And lastly, the 16S and NGS performance of MSA 1003, the 20-strain staggered proportion mix, was evaluated on PacBio's Sequel platform. Here you can see a consistent representation of the expected abundances, shown in gray, and the NGS and 16S results, shown in blue and yellow respectively. Additionally, the consistency between the 16S and shotgun data highlights the use of full amplicon 16S sequencing and how it eliminates the issues with 16S primers and conserved regions that I discussed earlier. And lastly, data analysis. The intent of this slide is to show the variability in data when the same sample set is analyzed with different commercially or publicly available platforms. You can see in the first graph where we show the results using the short read sequencing data of MSA 1005. The result with NIAID's Nephili showed slightly less reads for S- compared to one codex's analysis, which showed fairly even abundances. In the second figure, we compared the long-read sequencing data for MSA 1006. We were very pleased with the results obtained when the samples were analyzed with one codex's database. However, ONT's epidemiology showed an underrepresentation of some of the strains. And again, a similar situation was observed when we analyzed the data from our Mycobiome genomic mix with 2 different databases. Here, you can see the data was reproducible between replicates. However, when the same data set was analyzed with a second database, 3 of the 10 components were either not or barely identified. As you can see, bioinformatics analysis of microbiome samples can be challenging and can contribute to incorrect conclusions. Variables such as read quality, logarithms, and of course the quality and comprehensiveness of a database can contribute to inadvertent biases. Because of these well-documented issues, many of the microbiome products that I've discussed earlier come bundled with a data analysis solution. Select products come with a customized module on onecodex.com. The modules take into account each component, their respective genome size, 16S ribosomal RNA copy number, and published reference sequence. Customers select the genomic or whole cell icon, and then choose 16S or shotgun sequencing, select the correct module according to their product number, and then drag or upload their FASTQ file for analysis. After a brief period, OneCodex will return an analysis report which looks like this. Users will be provided with a scorecard such as the one shown here. All true positives were identified. That is, organisms that are known to exist in the mix. Any false positives, organisms that were detected but are not supposed to be in the mix, and of course relative abundance values. I would also like to mention that as of June 2019, customers will no longer need to enter in coupon codes to use the ATCC microbiome modules with ATCC Microbiome Standards. Customers can simply navigate through the website as I previously described and upload or drag and drop their FASTQ files to begin analysis. Here is a summary of the different standards that ATCC currently offers, and we are continually adding more to the collection. What I want you to take away from this presentation is that regardless of what type of microbiome research is being performed, one thing is abundantly clear. Standardization is essential. It is only with authenticated standards and optimized workflows that scientists can be able to achieve a clear understanding of the data and more specifically, how the human microbiome changes and develops over time, ultimately relating back to a deeper knowledge of human physiology. If you'd like to learn more about our collection or view our application data, please visit us online at www.atcc.org/microbiome. In conclusion, I hope I've demonstrated how the use of standards in all areas of research is absolutely essential. The inherent differences and complexities within each step of a given workflow can compound the number of biases introduced throughout the process, which can lead to an inaccurate analysis and incorrect conclusions. This is why standardization at every step of a workflow is so important, and also why scientists everywhere should be challenging their scientific peers to incorporate physical laboratory standards into their microbiome workflows to establish the validity, accuracy, and reproducibility of the data that they use to support new discoveries and technologies. And thank you to a great team, both internally and to all our external collaborators. And thank you to our customers and all of you for joining us today. And with that, I'm happy to take any questions.
Well, thank you, Briana. In just a few moments, we will begin our Q&A session. Please use the chat function available through the webinar program to submit your questions. The recorded webinar presentation will be available on demand on the ATCC website at www.atcc.org. All right, and we've just been joined by product line business specialists from ATCC, Steve Budd and Kyle Young. They'll help us answer some questions. Our first question that came in, it's around extraction kits, and I'll shoot this one over to Briana. For our first question, is it possible to know which extraction kit was used by ATCC to validate the results that were obtained?
That's a really good question. Yeah, thanks, Brian. We are often asked about extraction kits, and as I mentioned in the briefing, there is no perfect extraction kit for microbiome samples. We've had a lot of good luck with some of the commercially available kits from specifically QIAGEN. Their DNA Easy Powerlyzer is a nice kit to use, but typically speaking, there is no one perfect kit.
Okay, good answer, good answer. Now, here's an interesting question around recovery. Should we expect a whole cell mock community to have the same recovery as a genomic mock community? And just as kind of a follow-up to that, do you think that this might be subjected to the bias of DNA extraction procedures or kits?
So for the first part of that question, no, you're not going to get the exact recovery from a genomic mix that you get from a whole cell mix. The whole cell mixes will go through a bias that's not an issue with the genomic mix, because as I mentioned in the presentation, the genomic mixes are prepared. Each individual component is extracted using the most appropriate method for that particular organism. So you're removing that extraction bias with the genomic material. You will experience that bias with the whole cell. And then as the second part of the question was subject to the DNA extraction, yes.
Right,
so
right.
You will definitely see some type of bias with the whole cell. It's just an inherent nature of microbiome extraction.
Okay, I've got another question here. This is about, I guess, product design rationale. And so this attendee wants to know what was the rationale between 10 versus 20 strains, or even versus staggered mixes of the microbiome reference standard.
Oh, sure. That's a good question too. So with any microbiome, the more organisms in a microbiome, obviously the more complex the mix, the harder it is to detect everything in that mix. So that's just the rationale. You would start with, if you were just starting out, you might want to start with a microbiome mix that had lower number of organisms, maybe an even concentration. You're setting yourself up for success. You should be able to detect everything there in equal abundance. The more there are in the mix, obviously the harder it gets. And then some of them are quite challenging. 1003, for example, has like varying degrees of abundance from 0.02%, I believe, up to 18%. So that is our most challenging mix, the 1003. You've got your 20 organisms and they are in staggered concentration. So can you A, detect everything in that mix, and B, can you detect them in the abundance that we're telling you that they are there in?
Okay, great. Here's another question. And this is around species. So are there microbiome standards for mouse gut microbiome? And as sort of a follow-up, are the standards only relevant for humans? And this question, Briana, you can answer, and you might have something to add around this, Kyle.
So Kyle, I'll go first. So the gut microbiome that I mentioned earlier is specific to the human gut microbiome. That doesn't mean that you can't use it for NGS applications. Any of these are ideal for assay validation or limit of detection testing.
So I would just reiterate Briana's answer that these are intended for NGS. So they should be usable for whatever assay development you're working on, regardless of your end sample usage.
And actually, I did want to add something to that. If there's anything that customers are interested in that they're not seeing here, please let us know. You can just send a message through on this webinar. We'll be happy to address those.
Great answer. I'm gonna boot this one over to Steve. Do you offer any bacteriophage Virome standards?
We currently do not offer that as a product now, but we have noted that it is very interesting. I don't have an answer as to whether this will be available in the future anytime soon, but I will admit, yeah, this is something that we're aware of, and it is an interesting thing to look at.
Okay, good. Here's sort of a general question, I think, around biosafety that the microbiologists in this room could answer. What level of biosafety lab is required to work with bacterial and fungal mixes? Now, I guess we could answer this question, A, as far as the whole cell mixes goes, and B, the DNAs.
So on the product sheet, it's identified on the website what biosafety level it is, and labs have to have that biosafety level on record with ATCC in order to order the product.
Okay.
So the genomic mixes, for example, like MSA 1003, just check on the website. It'll tell you what the biosafety level is. Just verify that that's what your lab is certified to handle, and then we'll check it with our records. But we cannot ship material, obviously, if you're not on record with us having those credentials.
Okay, good. And that's generally true for any ATCC product, actually. So that was a good thing to bring up. Now, here's an interesting question. What is the best way to minimize contamination when doing microbiome analysis of low biomass tissue samples?
So I hate to answer a question with a question, and I apologize. Do you mean contamination during library preparation or data analysis?
We can come back to that if the asker will follow up. I will be looking for that. And we can sort of jump to the next question that's come in, and that's, what is the recommended concentration of genomic mix you recommend for consistent results?
So the genomic mixes, it will obviously vary on your platform that you're using. So when the genomic mixes come in, they come in a frozen format. So you should do either picogreen analysis or qubit, some other type of analysis to determine the concentration. For example, PacBio has a much different concentration requirement than Nanopore or the Illumina MySeq. So that's really going to depend on the platform that you're using.
All right, so you got to do a bunch of empirical experimentation to figure that out.
Well, it's different per platform, obviously. So just find out your concentration and then move forward with that.
Okay, good. Now, in the case of the 16S ribosomal RNA, which V region can represent the gut microbiome up to species level. And then as a follow-up, also mention the region which can be easily analyzed by QIIME2.
So we don't do species level for 16S. It's just, we prefer NGS. I would recommend doing NGS. We don't, like I mentioned earlier in the webinar, we don't recommend a particular assay. You would want to try V1, V2, V3, V4, and optimize it for your lab process. And was there a second part of that question?
Oh, the second part was also mention the region which can be easily analyzed by, I believe this is an instrument. It's QIIME2.
That's bioinformatics.
Okay.
You could use any of the bioinformatics platforms to do your analysis. Like I mentioned, we have the OneCodeX platforms, but that's a personal choice. Like kits or platforms, we don't recommend a specific bioinformatics platform either.
Okay, oh, it's Chime 2. That they followed up on. All right, cool. Thank you very much. Okay, well, we haven't had any, we actually just had a couple more questions come in. So one was the follow-up from that first question that I had asked about the best way to minimize contamination with the low biomass sample. And the clarification is, how can we minimize risk for contamination throughout the process of sample analysis, starting from the point of collection of tissue sample to freezing, banking in biorepository to extraction and sequencing? So this is like from the beginning to the end, just about.
That goes back to the very beginning of aseptic techniques. I mean, you should be doing all of your work in using your aseptic techniques, working in biosafety cabinets, handling samples in the appropriate manner.
Okay.
I refer back to we typically do and I'm sure it's saved, but we've done webinars about best practices in cell culture and aseptic techniques discovered in that.
That is true. That is true. You can look back through ATCC's webinar bank for more information about that. And also we have the culture guides, right? So we've got bacterial culture guide, or if you're working with protozoans, we actually have a protozoan culture guide, and of course a cell culture guide for mammalian cells. Okay, so then, wow, we got another one in. Do you recommend using standards only for assay development or including them with each extraction analysis set of samples?
I would definitely recommend using them as an extraction control, as a run-to-run control. You can use them as a daily run control. You want to show that your process is working every time you run it and you're recovering the same data that you would have expected to recover. And the only way that you can really reach that conclusion is by using that control every time you run.
Okay, good. We have another one. For environmental microbiology labs that are BSL-1, is there a whole cell mix that you are working to develop? I'd be really interested in being able to see what our current extraction methods best handle, but obviously being approved for BSL-2 would be pretty inconvenient.
That's a great question. We do not have any whole cells that are currently BSL-1, but that is definitely something that we can take into consideration.
Okay, and we'll give it just a moment to see if anyone has any last minute questions for us. All right, here we go. Do you have a microbiome and mycobiome standard mix?
We do not. We do not currently have mixed standards. We are working on some right now, but they are not currently available.
Okay.
But that doesn't mean, just to add a quick caveat, that does not mean that you can't take the current microbiome that's soon to launch and a standard microbiome and prepare your own.
All right, good answer. Well, at this time, we will conclude our Q&A session. I'd like to thank our panelists for the excellent presentation, and thank you everyone for attending this webinar presentation. And please join us for more webinars in the ATCC Excellence in Research webinar series. On September 12th, Balsam Shaki and I will present ATCC's new mouse STR service. And on September 26th, Kyle Young will present his talk on molecular standards. Thank you again, and have a great day.